Good morning, class. Let's go ahead and begin on today's lecture. I think this lecture is going to be around the same amount of time as yesterday, so it's not too long, but it's a little dense depending on some of the subject matter we discussed today. And then I will end up having some questions in the end today, but they're going to be more philosophical questions rather than actually like test questions. So today's class is going to be focused on prokaryotic viruses. So these are going to be the viruses that infect prokaryotes such as bacteria and archaea. So the general outline of today's presentation is going to be focused on the discovery of bacteriophages and then talking about some of the key structures of the virus, talking about its two different life cycles that it has, and some of the medical applications for bacteriophages. And then because we are talking about prokaryotes in this lecture, I also want to touch on antibiotics and resistance. I feel like that's an important topic that should be discussed in a class such as this. And then we're going to go ahead and talk about how bacteria are actually able to defend against bacteriophages, and then talk about this CRISPR-Cas9 system and designer babies and some of its implications for some of the ethical issues. So what I really want you to know from this lecture that you may or may not be tested on is know the key structures that I highlight of the bacteriophages. Because bacteria are so much more simple than us, the bacteriophages only have ribosomes to really hijack. So there's going to be two different life cycles that this particular virus has, but it's relatively simple. I would like you guys to know the medical applications of bacteriophages and antibiotics, and then know what the antibiotics target when it comes to discussing how it kills a bacteria. I also want you to know some of the main reasons why we're having such a big problem with this antibiotic resistance. And then know some of the key features of how the bacteria is able to defend against these viruses. And then I'm going to go a little bit in depth with this CRISPR-Cas9 system, but I really want you to understand like the big picture. What's the point of using the system and relating that to the implications of health and disease. So you guys have all seen this slide before. I showed this in lecture 1. So this is unique features that are highlighted for a bacteria. And so what I really want to for you guys to pay attention to is how simple it is and how little there is within the actual cytoplasm of the bacteria. And so the main thing that we're really going to be focused on that the virus is going to use are the ribosomes. So again, this lecture is going to be focused on viruses that are able to infect bacteria. So again, I mention this every time I show somebody that is famous, that has just made some kind of significant discovery. I'm never going to quiz you on this stuff, but it is important to give credit where credit is due. And so this is Felix De Jerel, and he is a French microbiologist. And so back in the day, his main focus was studying bacillary dysentery. So basically a bacteria that caused diarrhea. And what he noticed in some of the patients was that they were able to get better on their own. And he was wondering, well, how could this be? And so what he did was he took a sample from these patients and then basically filtered it out, where you basically took out all of the bacteria and the bacteria components, and then all you were left with was this solution. And basically all that was left in there, what he didn't know at the time, was the bacteriophages. And so what he ended up doing experimentally was taking some of that purified filtrate and adding it to living bacteria cultures. And then he ended up noticing that the bacteria were dying, even just with a small amount from that, the original filtrate, the bacteria were constantly dying. He repeated this experiment 50 times and always saw the same thing. And so he ended up concluding that there was some new biological agent that was killing the bacteria. And he's the one that's credited for naming bacteriophages, which just means bacteria eater. And this was discovered in 1917. There are many different kinds of bacteriophages, and they all have different genomes. So the ones I'm probably going to be focused on more, the DNA virus ones. But it is important to note that there are bacteriophages that do have RNA viruses. And really the whole point of these viruses is to kill off bacteria. There's always been this symbiotic relationship with bacteriophages and bacteria kind of fighting against each other. And this was long before we ever existed. I'm not going to expect you to know all of these different names. I'm just going to, just generalize bacteriophages for this lecture, as there are a few different types. So where do you actually find bacteriophages? They actually live wherever bacteria is at. So they can be found in soil, and here I have what it's estimated to have. So there's 10 to the 10th power amount of bacteriophages that live per gram of soil, so that's quite a bit. And then they also are found in seawater, and there's estimated to be 10 to 50 million phages per mil. Surprisingly, they're actually found in our body. We have bacteria that's on our skin and in our gut, and that bacteria is good, but you have bacteriophages that are also wanting to kill some of those or regulate the bacteria so there isn't overgrowth. Here I have basically an image of a typical bacteriophage structure. So, the bacteriophages and phage are used interchangeably, but they mean the same thing. And so their whole point is to infect bacteria. So bacteriophages do not infect animal or plant cells. It's only bacteria. So if I were to ask you a question, what do bacteriophages infect? Know that they are a virus that infect bacteria. There's a lot of students that get bacteriophages and bacteria confused, thinking that bacteriophages are a type of bacteria, but they're not. It's a virus. Like I mentioned earlier, they can have a DNA or RNA virus depending on the type. But what's interesting is they have these tail fibers that help facilitate bacterial entry. So these are what are going to recognize specific receptors that are on the bacteria's surface so it can actually bind to it successfully. There's also these spikes in base plate. And what these bacteriophages do is, instead of doing what we discussed yesterday where viruses will fuse their membranes with the host cell, bacteriophages basically poke a hole into the bacteria. And once that hole is made, this sheath, or other textbooks might call it a tail, will inject the genomic material into the bacteria cell. Like all viruses, once the viral genome is within the host cell, the viral genome will be able to hijack host cell machinery to go ahead and facilitate replication. So bacteriophages are relatively small and simple, but they have two different life cycles. So this first one I want to discuss is called the Lytic Cycle. And so another name for the Lytic Cycle is also known as the virulent or infectious cycle. And what this is going to end up causing is lysis of the cell, or it's going to cause the host cell to burst open. And this is different compared to, say, animal cells, where they just basically will replicate within the host cell and then, bud out and the cell is okay until the immune system comes and recognizes it. Well, bacteriophages, when they're in the lytic cycle, they actually end up killing the bacteria. So once the viral genome is actually in the bacteria cell, it's going to end up degrading all of the bacteria DNA. And so that's basically to have less competition, so its viral genome can go ahead and hijack the host ribosomes and then begin protein synthesis. And so this protein synthesis is basically going to start making different viral components. Once these viral components are made, they're going to be assembled, and they have their own machinery to be able to do this. They encode specific proteins that help with the assembly. Once there is enough of the phages assembled and made, there's going to be enzymes that actually break open this bacteria. And this is going to release anywhere from 100 to 200 phages. And then the whole cycle can go ahead and repeat, where it'll go ahead and start infecting other bacteria that may be nearby. The other cycle is known as the lysogenic cycle, or the non-virulent or temperate infection. And so this one isn't going to kill the cell. What it actually does is, once the genetic material is deposited into the bacteria, some of the viral sequences are going to encode for this enzyme called integrase. And so integrase, has the ACE again, it's an enzyme, is going to end up integrating some of the viral genome with the bacterial DNA. And so, excuse me, the viral DNA at this point, once it's fused with the bacterial DNA, is going to have the name prophage. And so this prophage can basically be copied passively when a bacteria cell is replicating, and it can actually pass it off to bacteria daughter cells. If the bacteriophage is in the lysogenic phase, it's just going to continue to stay in that phase. But once it starts to experience some kind of stress, such as UV light, there's not enough nutrients present for it to live, or some other kind of chemical stress, it's going to end up upregulating some of those key proteins, so it can enter the lytic cycle, so it can start assembling copies of itself and starts trying to get out of the host cell and infect others. So this process isn't perfect, and what ends up happening once the bacteriophage ends up undergoing some of this stress, once it's trying to rush out of the host cell, sometimes it might leave behind some DNA, or worse, it actually can uptake some of the host cell DNA. And again, this host cell DNA is the bacteria DNA. And sometimes this DNA can encode for antibiotic resistance genes. So basically genes that if you were to treat a bacteria with an antibiotic, it's not going to be able to kill it because it encodes for the specific antibiotic resistance sequence. And so if a bacteriophage ends up picking up some of that resistance genes and ends up surviving once it breaks out of the host cell, it can actually infect other bacteria and transmit some of these genes to the other bacteria populations. And this is one way how antibiotic resistance bacteria can form, but it's not the only way. And I'm going to touch on that in a bit. So back in the day, before antibiotics were even discovered, scientists were trying to figure out ways on how can we fight against bacterial infection, because during the early 1900s and before, a lot of people ended up succumbing to bacteria infections, and we didn't really have a good treatment option. And so before antibiotics were discovered, phages were being explored to be used as a way to kill off pathogenic bacteria. And so again, the bacteriophages weren't discovered until the late it was 1917 when it was discovered. So 10 years later, the first antibiotic, penicillin, was discovered by Alexander Fleming. And basically, because this antibiotic was discovered, and it was learned that these antibiotics could kill multiple species of bacteria, not just one, bacteriophages being used as a treatment to treat bacterial infections were forgotten. And so when Alexander Fleming first discovered penicillin, he ended up discovering this by noticing that a plate of agar, so basically nutrients for bacteria, he noticed the mold was growing, but he also noticed that wherever the mold was growing, bacteria were not growing anywhere near it. And then that's when he ended up learning that mold made this molecule penicillin to keep the bacteria at bay. So, there's this other, microorganism fighting. So before we had, viruses and bacteria fighting against each other. Now we also have fungus fighting against bacteria. And so basically a lot of our antibiotics are derived from purified molecules from different kinds of fungus species. And so back in the day, basically the way that they ended up making the penicillin was they ended up growing up the fungus penicillium and gave it basically some sugars so it can go ahead and grow and ferment. And then basically after there was enough of it, scientists were able to basically separate and purify the penicillin molecule from the mold. And then that was used for treatment for different bacterial infections. So antibiotic mechanisms all are dependent on the class of antibiotics. The whole point of them is to kill bacteria. It's important to note that they do not kill viruses or fungi. It's all meant to kill only bacteria. So if you were to get a question like what do antibiotics treat, you better know this answer, because a lot of doctors don't, and that's why we have resistance. And we'll get into that in the next slide. So basically, depending on the class of antibiotic, it's going to target different parts of the bacteria machinery. And so penicillin actually is used to inhibit cell wall synthesis. And the cell wall is really important to protect the inner workings of the bacteria. And so if you have a compromised cell membrane, it's going to be very weak. And if it's very weak, it's basically not going to grow very well, because the cell wall is important when it comes to cells needing to divide. Other classes of antibiotics can basically target protein synthesis. So I mentioned before that animal cells and bacteria cells both have ribosomes, but the ribosomes that are in bacteria are different than the ones in us. They have the same function where they are involved in protein translation, but they look different, or they encode for different things. And so the antibiotics that we have can specifically inhibit these particular ribosomes to prevent protein synthesis from occurring. And so if you don't have the protein, then you can't actually make the bacteria component. And then other classes of antibiotics will focus on actually preventing replication. So as we discussed in lecture 2, before any cell ends up dividing, it needs to replicate its DNA. And so if you don't replicate the DNA, then the bacteria can't divide properly. So know these key components that antibiotics inhibit when it comes to trying to treat against bacterial infections. So antibiotic resistance is a big thing. And before I get into that, I want you guys to see this video, and then I'll go ahead and expand on this, because I think it's kind of an important thing to be aware of if you guys haven't seen this video already. And let me know if you guys have trouble.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands. And at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So hopefully you guys found that video useful. So basically, we've had antibiotics for over 100 years at this point, but we are starting to develop antibiotic resistance for a couple of reasons. One of them is doctors are to blame, if I'm being honest, and it's sad to say this, but a lot of doctors will overprescribe antibiotics. So what ends up happening is they think that the antibiotic is going to treat a viral infection. So if you have the common cold, which I discussed yesterday, is a viral infection, they end up prescribing antibiotics for that, which is going to have no effects. And by treating patients like this, you're not going to only kill off the bad bacteria, but you're also going to kill off the good bacteria that exists within you. And so if you're killing off that, then you basically have this chance of bad bacteria basically over reproducing and then creating a big problem within the patient. The other, and it's not just doctors that are to blame, it's also the patients. So a lot of patients, if they are treated for bacterial infections, a lot of them don't finish their course of treatment. Typically, courses of treatment are around 7 days. And so I think on average, it's around 4 to 5 days that patients start to feel better. And so then they stop taking it. And so this takes the risk of developing one of those mutants because you haven't killed off all of the bacteria that are present. And so if you haven't done that, then you take that chance of when the bacteria are replicating, you take that chance of one of them basically developing one of those resistance genes by either uptaking it from a different bacteria or by them creating some kind of mutation within their genome that's going to end up encoding for the resistance. The other main source of why we're having such big issues with antibiotic resistance is it's overused in livestock. So basically 70% of the animals that we consume, they are being treated with antibiotics relative to humans, where of those antibiotics that are being used, only 30% of that is going to us. And so this isn't good because animals are a source of how both viruses and bacteria end up causing disease and transmitting it to humans. And so if you end up giving these animals antibiotics preemptively to prevent any kind of bacterial infection, you take a high chance risk of developing some of these mutants. And so bacteria, are going to become resistant to our first line of defense, and it's already begun. And according to the UN, it's estimated by 2050, there's going to be more people that die from antibiotic-resistant bacteria than cancer. And that's quite alarming because if you look at the causes of death, at least in the United States, heart disease is number 1 and cancer is number 2. Of those 10 that are on the top list, bacterial infections isn't on there. But in the next, couple decades, I'm sure we will end up seeing those numbers begin to rise, not only in the United States, but worldwide. And unless we find some kind of alternative. And so one alternative that scientists are re-looking at are phages. And so as more and more bacteria become resistant to antibiotics, we're thinking that a phage might be the solution to this. And I included this quote here because I really enjoyed it, and I feel like this potentially could be the future. Traditional antibiotics are losing their edge, and the phage are the new weapons in our battle against bacteria. And so we have to also consider, some of the main drawbacks to this, though, and why it's not really being used at this point. Biologically, bacteriophages only are able to kill certain species of bacteria. It doesn't kill off all types of bacteria compared to antibiotics, which one drug can kill many species. And so unless we're able to somehow genetically modify bacteriophages where they can do this, at this point, we're very limited on the kinds of phages that we're able to use. Financially, you can't patent phages. They're a biological substance. And so if you can't patent something, then Big Pharma has no reason to invest in it. The only way Big Pharma will ever invest in some kind of phage or phage therapy or related molecules is if you're able to synthesize them and they're able to market them. If they're able to synthesize their own drug or molecule that can fight against bacteria, then they will do so. Wait, so what is in antibiotics? So you're asking what is in antibiotics nowadays? Antibiotics are the same thing as they've always been. It's just bacteria have mutated where they're no longer being susceptible to death from the drug, if that makes sense. So if you look at the structure of some of the antibiotics, they're similar to nucleotides. And so with bacteria, basically they are able to, they end up, and instead of like using an A, they end up using the antibiotic and incorporating that into the DNA. And so when that gets translated or something, it's not able to read it properly. And so you're not able to synthesize the proteins. But a lot of bacteria are basically not even looking at the antibiotic anymore because they have certain genes that encode them that they don't even consider it anymore, if that makes sense. So one of these molecules that might be a potential candidate are these lysins. And so lysins are enzymes that bacteriophages use to break out of the host cell. So after the assembly is all done and it's ready to lyse or break open the cell, it uses this enzyme lysin to do so. And so we have a micrograph image of this enzyme being worked to release all of these bacteriophages. And so scientists have now been able to isolate and purify some lysins from the phages. And so here we have an image of scientists treating bacteria with lysin to kill off the bacteria. And so instead of the lysin working from the inside out to burst the bacteria membrane, it's actually being used from the outside. And then once it does that, it's basically like a balloon. Once you make a hole in the balloon, there's so much pressure that is within the actual cell, it ends up just releasing all of the cytoplasm and some of the cargo that is present within the bacteria. So can bacteria fight against phages? And if you didn't know this, bacteria actually have an immune system. The first time that I found this out, it was just crazy to me. It's not as complicated as how we have it. It's a little different. And so I'm going to kind of walk you through a little bit of this because I think it's very important to understand this. And so the bacterial chromosome has this Crispr locus. And so basically within this locus or sequence of genes is going to be this clustered regulatory interspace short palindromic repeat. So that's what it stands for. That's probably the only time you're going to hear me say that. Everyone refers to it as CRISPR. And so within this segment of DNA, there's two important areas that I want you guys to know that are really important when it comes to facilitating this immunity against bacteriophages. And that's going to be the CRISPR array and the Cas genes. And so within the CRISPR array, that's where segments of viral sequences are present. And so going back to the lysogenic cycle of the bacteriophages, when it's integrating its genome into the bacteria, basically one of the regions that ends up getting fused with that viral genome is this CRISPR array. And other segments of viral DNA are when the bacteria were actually able to successfully defend against the virus. And I'll go and explain the steps of how this works in the next slide. But I think to understand some of these terms and key genes and proteins are going to be critical to understand the actual process of this. And so the other main genes that are important to know are these Cas proteins. And basically they encode for Cas proteins, and there's a few different kinds of them. The only one I'm going to have you guys really know is Cas9, which I'll get into in a couple of slides. But what these proteins do are they're a type of nuclease or an enzyme that are able to cut genetic materials such as RNA or DNA. And so when we actually look at how bacteria are able to defend against these phages, we basically have these steps. So when the bacteriophage is depositing its genetic material and the lysogenic cycle is occurring, some of its segment is going to end up being placed within this Crispr array segment. And when the bacteria recognizes that there's an infection going on, there's a whole signaling cascade that we're not going to get into. I'm going to keep it as simple as possible, that basically upregulates or causes the production of these Cas proteins. And again, these are going to be the nucleases that are going to be used to fragment the bacteriophage's DNA. Within this Crispr array, there's a whole process that ends up occurring where all this viral DNA will end up being basically transcribed into these, they're called tracer RNAs, and you don't need to know this, is just more for me explaining. But what you do need to know is once this Crispr array is, transcription ends up happening and making these sequences present, they're going to end up getting broken up into these single guide RNAs. And basically, these single guide RNAs are going to end up fusing with specific Cas proteins. And so what the single guide RNA is going to do is, just like the name implied, it's going to guide the Cas proteins to the genetic material that's being deposited into the bacteria. So what it does is this complex will end up scanning that DNA that foreign DNA that's being deposited into its cell. And then once it binds to its complementary sequence, it's going to end up binding, and then these enzymes are basically going to end up fragmenting the viral genome. And so if you fragment the viral genome, then you're not able to actually make productive virus. And what's more fascinating about this is the bacteria is able to take some of those fragments and add them to the Crispr array. So now it has even more immunity or memory against the next set of viral infections. Let's see, question. Much more difficult for bacteria to develop an immunity. I figure then that if it is much more difficult for bacteria to develop an immunity to phages because they have a physical damage aspect where they break the cell wall, which makes me wonder, do phages affect plant cells? Not to my knowledge. From my knowledge, the only bacteriophages only infect prokaryotic cells. So it's only unicellular organisms such as different species of bacteria and archaea. And so I think if you're referring to like plant cells dying, I think it's just because of how, I guess it just depends on the replication cycle for that particular virus. And it wouldn't surprise me if plants have some kind of immune system as well that might be leading to some of their death. But yeah, so bacteriophages only infect unicellular organisms. So a little bit more about the CRISPR-Cas9 system. So I don't know how many of you guys have heard of it, but it's a very important system to know because I believe it's going to be used in humans in the future, and it actually already has. and I'll end up getting into that in a couple of slides. So basically, what humans are really good at are exploiting mechanisms from other species. And so we ended up doing this with the bacteria's immune system. And so now we have this ability to knock in or out genes. And so before, to basically create mutations, we would only be able to stop gene production partially. We were never able to do this fully before until this system came about. And the system's relatively new. It's, even though it's existed in bacteria for however long bacteria have been around, humans have only really been working with this in like the last decade. And so I'm not going to have you guys know this whole exact like depth, but what I do want you to know is that the CRISPR-Cas9 has the ability to knock in or out genes, and their mechanisms are quite simple. So like I said with the Cas proteins, basically they're going to end up, they're a nuclease, so they're going to end up creating double-stranded breaks, or they're going to end up causing breaks within genetic material. And this single guide RNA is going to direct where that cut ends up happening. And so if I'm interested in knocking out a gene, basically I'm going to design a guide RNA that's specifically for that gene of interest. And then once this whole complex forms, basically two things can happen. It's all dependent on what's present. So for the knockout mechanism, basically what's going to end up happening is the double-stranded break is going to occur, but because we're more complex than a virus or a bacteria, we have host cell machinery that's going to facilitate that repair. But that repair is going to end up creating these insertion deletion mutations. And what's going to end up happening is a frame shift's going to occur. And so if you remember a couple of lectures ago when I was referring to the different kinds of mutations that occur, this CRISPR-Cas9 system basically creates a mutation where it'll end up either the polymerases will add or take out extra nucleotides, but that's going to shift the sequence completely. When that sequence is shifted, it's going to end up creating this premature stop codon. And if you remember from that lecture, the stop codon basically tells the transcription and translation machinery to stop synthesizing protein. And so if you end up having that stop codon too early on and the protein's not fully formed, then the protein's going to end up getting degraded because it can't be folded properly or it's not fully functional. And then for the, and so, it'd be a good idea to review what kind of mutation this is again, because you might get a question on that. So, I want you to understand that. For the knock-in mechanism, it's going to be the same where you create that double-stranded break, but you also need a couple of other things that are present. But the main thing that you really need to know is whatever gene you're trying to knock in or repair, that sequence needs to be available for the host machinery to be able to insert it into the proper location. If that sequence isn't present, then this non-homologous end joining or this indel mutation process is going to end up happening. But if it is present, then basically that sequence will end up being put into wherever the guide RNA told the nuclease to cut. So here is a close-up of this complex assembled onto the DNA. So here in yellow, we have our Cas9 protein. So I mentioned before, bacteria have different kinds of Cas proteins, and the different proteins have different enzymatic activities. So sometimes some of them will create like only, it'll cut one of the strands, whereas the Cas9 protein will actually cut both strands in the location that it's directed to cut. And then with the single guide RNA, there's actually two parts to it. There's this complementary sequence that scientists are able to design that are going to be complementary to whatever your gene target is. And then you have this scaffold sequence. And basically this scaffold sequence is going to hold the complex together. So connecting the enzyme and the complementary sequence. So in terms of knowing the parts of this, these are the key things that I'm going to want you to know. So how can we use this system for our own gains? And so there are, a couple of thoughts to what scientists and physicians are trying to do. And so one way that scientists are trying to utilize the CRISPR technology is by using it to fight against cancer. And, there's different, depending on, the mechanism that you're trying to target, this CRISPR system can be used differently. And so here I have basically CRISPRing in or editing T cells, which I'll end up getting into next week when we're talking about immunology. But for right now, what you want to know is that T cells are really important for killing cancer cells. And a lot of the time, cancer cells express certain proteins that inhibit T cell function. And also, T cells recognize specific proteins on cancer cells. And so if you have cancer cells that are inhibiting its function, then you're basically not going to kill off the cancer. And so what scientists have been able to do is take T cells from a patient that has cancer, genetically modify it using the CRISPR technology to basically be able to recognize the cancer more efficiently. And so they ended up using this receptor depending on whatever cancer they were targeting. And so basically when you are able to make this receptor more efficient at recognizing the cancer, then you're, and once, there's going to be a whole screening process to make sure that the T cells are healthy and looking good. Then you're going to be able to put that back into the patient. And then the thought behind this is that the T cells are going to be more efficient at recognizing the cancer cells and then killing it, because T cells are important for killing specific things within the body. So usually it's due to infection, but it's also from cancer. Then we could also potentially use this to cure genetic mutations. So going back to that mutation slide from whatever the lecture was, there's over 3,000 genetic diseases that are caused by a single point mutation, and these single point mutations can have varying degrees of effects. And it's dependent on what the gene encodes and what the protein ends up getting made from that that mutation. And so a single point mutation can cause color blindness, so that's very mild, and then all the way up to Huntington's disease, where that's going to create problems within neurons. And so scientists are in the process of trying to genetically modify some of the Cas proteins so it can just take that one, instead of creating that double-stranded break, they're trying to take that one messed up point mutation or one nucleotide and put in the right one, so it can go ahead and basically fix the genetic problem. Is CRISPR exclusive to bacteria? So yes, the CRISPR system originated from bacteria. That's where it's going to be found. It's not found in any other species. In order to use it in us, we actually, because bacteria are so simple, we're able to basically take the sequence that encodes for the Cas protein, and our own host cell machinery, once it's in the cell, is able to actually make that protein. And then, yeah, so hopefully that answered your questions. Yes, it's specifically in bacteria, but humans can make it ourselves and put it into us. And then the other the other I guess use for this technology is making designer babies. And when I refer to designer babies, I'm not referring to a baby that's dressed in designer clothing. I'm referring to genetically modified babies. And if you didn't know, The first CRISPR babies were born in October 2018 in China. And so what the scientist was trying to do is basically modify the embryos so they make them less susceptible to HIV. And so we didn't talk too much about it. I think I mentioned it in the intro virology slide. But in order for HIV to get into a host cell, there are two receptors that it needs, CCR5, which is listed here, and CXCR4. And so what this scientist did was basically mutate the CCR5 gene so that protein was non-productive. And so if it's non-productive, then that means it's not going to be present. And so this virus needs both of those receptors in order to get into the actual the host cell. So if it's not present, then you really reduce the chances of it ever getting into the cell, and you're technically immune. However, there was a lot of ethical dilemmas in all of this, and the scientist is now serving 3 years in prison with a $430,000 fine. So we can design RNA or DNA for any protein we want, and all we need to do is get the sequence. For the most part, yes, it's just dependent on how big that sequence is. So we're able to use this for from bacteria because their sequences are so small. Now, for us, a lot of the proteins that we have are very large, and so trying to get a very large sequence into a host cell, even though to the naked eye we can't see, for it to actually get into the host cell, it's very difficult. So it's all dependent on the size of the sequence. And so with the Cas, the sequence that encodes for the Cas proteins, it's very small. So as long as you can get that sequence into a host cell, the host cell machinery can go ahead and synthesize it. I hope you guys found a lot of this lecture interesting, and I feel like it's really important to discuss some of these things, because if you haven't been aware of a lot of these new technologies that are being created, you're going to eventually hear about them, because if I'm not mistaken, this is going to be the future. And so what I would like you guys to all do is basically go out into breakout groups and then discuss some of these questions. And, I hope, that there is no right answer to these. I just want to see what people are thinking. And then I'll give you guys about 10 minutes to do this. And then I'll end up polling you guys on some of these questions. And I'm hoping that some of you guys will be brave enough or not shy to, state your opinion, why you support or against some of this technology, whatever the question is. Because I think some of these discussions need to be had, and I think it'd be good to start this in a learning environment. So I'm gonna go ahead and stop sharing this right now. I think you guys have all access to this, and then I'm gonna put you guys in breakout groups. 1 second. Yeah. Oh, and then also, if you guys are staying in this breakout room, in this main room, which some of you guys do, it's telling me that you're not actually participating or even at your computer. You're not going to get your participation points for the day, so please actually participate, because that's what those points are for. It's not enough that you just come to class, it's to participate. So I'll give you guys 10 minutes, and hopefully all you guys have some fun. So the people that are present, are you guys not at your computers, or what? Are you guys having trouble transitioning? Adrian, are you having some issues? All right, let me see if I can move you to a room where there's only 2 people. 1 second. Amber, are you having issues moving to a room? Oh, no worries. Do you remember what, actually, were you in room 6 before? Oh, 22, okay. Oh, there's nobody in that room. I'm gonna move you to room 6. Mhm. Give it a couple more seconds before everybody comes back. All right, I think that's everybody. So hopefully you guys had some good discussions. So for the first question, I want to know for the class, are you guys supporters for the technology? you guys a couple more seconds. 3, 2, 1. All right. It seems like more people are more passive than anything. If you guys don't mind, can I have a proponent for each one? If somebody could say why they support it, why they don't, and why they're, indifferent. Oh, the question was, are you a supporter of the CRISPR-Cas9 technology? Anybody?
Yeah, I am. I think I would say that I'm a supporter of it because this is like, I guess, in regards to evolution, this is like humans pulling the trick card on nature. It's like, well, you don't want to evolve us, we will. And we can really do a lot of good with it. And so I think if we use it correctly and we develop the technology well enough, we could pretty much change the course of humanity forever.
Yeah, I agree. I'm also a supporter for it, and I like all the points that you touched on for sure. Anybody that's against it and do they want to share? Don't worry, you're not going to be viewed as like an anti-vaxxer or anything. Let's see. Somebody wrote, I am neutral because I think it depends on what you're using technology for sure. Somebody had a raised hand, but it went away. Go ahead and unmute yourself.
Hey, Professor, can you hear me?
Yeah.
Yeah, no, I think it's a great question, and definitely something like a lot of us I'm sure would love to talk about. I am on the more neutral side, and that's because I understand like the good that can come from it, but I also recognize the clear bad that is definitely possible with that kind of research. So that is why I'm in the middle.
For sure. No, and I agree, because I feel like if we look at history, like even though I'm a scientist by training, I love history. I learn from it, and I feel like whenever we develop something for the benefit of humanity, we always somehow manage to use it to hurt our own people. And so yes, I see how it could be used for bad. Somebody said, I'm neutral, but imagine not having a genetically bad eyesight, all the money that could be saved. Yeah, definitely. All right. So that kind of answers one of the questions. Okay, let's see. These are good discussions. Yeah, thank you guys for sharing. I appreciate it. Okay, I'm gonna go ahead and stop sharing this. And then the other question I would like to ask is, would you ever use this on yourself or your child? Give you guys 5 more seconds. All right, basically everybody. Yeah, you guys are all split 3 ways. Can I go ahead and have somebody share? Let's see some stuff in the chat. That's a tough one. Yeah. So say like your kid had a disease or something, would you use it, or would you use it more for vanity purposes? I like to hear people's opinions.
Professor?
Yeah.
I think personally, I would use it if it was to prevent or decrease the chance of like some type of disease. Just to like prevent having to worry about that or spending the money on that or whatnot. I don't think I would use it for any like superficial like vanity stuff, because I guess that's kind of more changing the way you're meant to be. And I don't know, I feel like it's very like superficial and type of shallow. Obviously, like not like judging anyone who would do that, but I would definitely use it in order to decrease chances of disease or any harmful type of stuff for sure.
Yeah, thank you for sharing. Anybody else?
I agree with what Sergio said too. That you would probably be good for using it for preventing diseases or all those things. And like from what I've heard of this system, it works better when it's applied to babies that haven't been born yet. So in this case, it would be the parents that would get to decide if they wanted the baby to have like specific physical features. And I think that's a very like not morally correct, because that baby is also another person and they don't get any say in what they're gonna look like and whether they're gonna like it. So I think it's better not to mess with that part of it.
Thank you. No, that's a very good point. Yeah, that's, they don't have a say, so how can they, what right that you're almost stripping the fetus's rights off the back. So, it's, yeah, very good point. Anybody else before I move on? I feel like this brings out the question, who would you be able to use this? If you have more money, you're allowed to have a more perfect baby. Exactly. Yeah. And I guess I would also use it on my child, but only really if it was detrimental to my child's health. Yes. Okay, good. Yeah. And to kind of elaborate on that, I agree with all of what has been said. Me personally, I would use it on my child, but only if they had some kind of disease. I feel like it's unethical for vanity reasons. But unfortunately, I think once this technology is perfected, and we understand genes better, I do think that some of the superficial stuff is going to occur. And unfortunately, it's going to be the rich that are going to be able to afford this. And I think that's going to lead to more socioeconomic divide than we already have. Right now, it's race is used to break people apart, but we have to pay attention to it. It really comes down to money, rich versus poor. And then when you throw in genetics into the mix, it's just going to be even, exacerbate that chasm. Yes, this would bring up a class issue. And I think more down the line, it could create a type of division fight between, yeah, exactly, genetically engineered humans. A lot of someone said it sounds like a sci-fi movie, but a lot of sci-fi movies become reality, and it's I think it's going to happen probably in our lifetime, if I'm being honest. It just we think about it. We've developed antibiotics like not that long ago. This last decade, last century, we've really, our technology has exponentially increased. And so in the next 100 years or next 500 years, it's crazy what humanity is going to be able to accomplish, assuming we don't kill ourselves. All right, so the last question I do want to ask is, should the Chinese scientists have gone to jail? Let me relaunch this. All right, more or less everybody is done. So share results. Most people seem to be neutral about this. Can I ask why people are for each of these?
I voted yes, that he should have went to jail, but not because I'm against what he did, I guess, but mostly because this technology is very experimental at the moment. Mm-hmm. And so I think having applied it to a human in this phase, it was very not a very what's it called? The word careless in this sense, right? They shouldn't have done it yet because I think it's very too early. But other than that, I think I'm for the technology. But in this case, I think the scientist was too quick to do this.
Definitely. I hear you for sure. And it's, I wonder, because these babies were born, I'm wondering how they're going to be in like the coming decades. Are we going to actually, they're basically the first guinea pigs, if we're really putting it in perspective. What I find most concerning about, I guess, what they did with this technology was they knocked out a receptor that's very important for immune cells to communicate and migrate. CCR5 is a chemokine receptor. So chemokine receptors, basically think of it as like being able to smell perfume. So if you spray perfume somewhere out there, and then the odor kind of trickles out, the immune cell is able to go and follow to see where it's most concentrated at. And so if you get rid of that receptor, that's going to have some consequences when it comes to being able to fight off infection. So I'm interested to see what's going to end up happening in the future with this. Somebody said, I think more testing should have been done before that. Definitely. They said that they tested them and they didn't have any issues, but I don't know how rigorous that testing was. Anybody else want to say any last comments before I cover my last 2 slides? No. All right. Thank you guys all for participating. I think this was great discussion. I hope you guys enjoy your discussions within the breakout groups. But yes, these are things that we need to be talking about now, because this technology, I think, is going to be available to everybody in the future, not just a select few. But I think a select few are going to be able to afford it for a time until it gets covered by healthcare. All right. So there are drawbacks to the CRISPR system, and I've actually used this technology in my last lab I worked in. I was actually trying to basically knock out key regulatory genes in macrophages. And then what I would do is I would infect them with the virus and then see what ends up happening. And, you knock out some genes, and that's going to end up leading to, higher rates of infection. So, we were able to say that gene actually was important for that. However, with the single guide RNAs, they're not perfect. There's a lot of off-target effects. So what this means is the sequence, it's only about 20-25 nucleotides long. And so a lot of the sequences, they don't need to match 100%. And so if the sequence is 80% similar to another gene that it doesn't match whatever you're trying to do, the nuclease or the single guide RNA will guide it to that other area and actually cause breakages there. And that might create problems down the stream. And when I was trying to use the system, I experienced this. I was able to get all of this into the cell, but I wasn't able to actually knock out my gene of interest. It was cutting other places, and you don't want that. And then in terms of, I guess, physiologically, getting the CRISPR into cells is quite difficult. Most scientists are able to do this in a petri dish because they have, specific, they're called lipids, basically, or vesicles that are able to basically help facilitate that entry into the cell. But when you're trying to put that into a human that's already living, it's quite difficult because I think the technology and the sequences are very unstable. So you take the risk of it degrading while it's in circulation in the blood, because it's going to be something injected. You can't eat this. Or your immune system is going to end up recognizing it as foreign and getting rid of it. So the reason why the Chinese scientist was able to do this with the embryos is because they were at a cell stage. And when they're at that cell stage, it's very easy. We have technology that we've had for a long time to be able to get that those sequences in. And then here I just have a couple of pros and cons. Of course, I'm not going to quiz you on any of this, but it's important to really weigh out both of these when it's considering, is this technology really good? And I think in the end, if laws are set in place before it becomes actually a common practice being put into humans, I think you're going to have a huge benefit for therapies. But like I was saying before, humanity has a very ugly and dark side, and it's not going to surprise me if this technology ends up being used for bad in the future. I'm going to go ahead and end this. If you guys have any questions, let me know. I'm going to go ahead and throw in the sign-in sheet into the chat. And you should be able to edit it. If you can't, let me know. But thank you guys for coming. And again, the midterm is next week, next Tuesday. So please, if you have any questions, if there's any concepts from the learning objectives that you don't understand, let me know, okay? I have office hours on Thursday. I can stay a little bit longer right now. I do have a meeting at 11. So If you have any questions, ask before you end up missing it on the exam.